I'm the Education Director with NOFA-Mass, Northeast Organic Farming Association, Massachusetts chapter, and I am also our Soil Carbon Program Coordinator and a soil health consultant. And I am out here today at Caro Community Farm in Woodbridge, Connecticut. And what we're doing today is demonstrating a soil health assessment, or soil carbon proxy test, as we call it at NOFA. So before you begin to conduct your full tests, if you're a technician doing this test for a farmer, you want to conduct an interview with the farmer to get history of their farming practices, such as tillage, mulching, cover cropping, and the last few crops that were in this field, things like what fertilizers they're using if they're applying compost. And you want to record things like the weather and the temperature, obviously the date, because you want to conduct this test at the same time every year, and draw your map to where the test is taking place. At that point, you can move on to begin your tests. So there's two ways to use these tests if you're doing your own soil health assessment for your farm. One is to do a comparison test, so that would be to test two different fields or two different areas in your field or multiple areas in your field. In that case, you'd want to choose different locations within your field that you know have different soil conditions, such as higher, lower, rockier, less rocky, wetter, not as wet, and compare those. If you're going to be using this method for tracking change over time to look at your management practice and how that's affecting your soil health from year to year, you would just choose one location in your field that's representative of your field and look at that same exact spot every year, because as we know, soil conditions are variable, so you want to choose one spot and compare that over time. The materials for doing this test are really quite simple. It's just a long surveyor's tape. I had a 150-foot tape. I switched to a 300-foot tape for different farms, because what you really want to be able to do is measure from a permanent landmark. So on this farm, what I've done is looked to the absolute corner of the farm. I'm going to draw a map with cardinal directions, and I've counted from that corner 8 posts. So when I record the location for this spot, it'll be count 8 posts up from the southwest corner, and then measure 115 feet and 6 inches to the center of the test location at 185 degrees on my cell phone compass. So that's basically the directions I'll record for myself. There's also some water mains nearby, just simple metal poles that indicate where the water sources are. So if you wanted to get really fancy, you could use your compass and actually also in your surveyor's tape and triangulate from those spots if you wanted to get really precise. You can also use GPS apps on your phone, but they're generally not as precise as these directions you can make for yourself. The materials that I've used for this, like I mentioned, a 300-foot surveyor's tape. This is just simple PVC pipe cut to a specific length and joined with a simple coupler kit that you can get at Home Depot or your local hardware store. And the goal is to make a 30-inch diameter hoop. This just allows you to better estimate percentages of cover. And the hoop is just to sort of help you focus on a specific area of a contained amount of space. And what the goal is to estimate the percentage of living cover, residue, or dead cover, and bare soil in the hoop. And the test is conducted from a standing position looking down for estimates. So we're not getting down here and like moving the plants around to kind of like look at the bare soil. This is just a simple surface observation. So from my perspective, standing here, what I'm seeing is about 5% bare soil. Very little what I would call residue, less than 2%, maybe closer to 1% of residue, just kind of dead plants. And mostly living plants, which is really good. There's actually companies now that work for farmers by conducting satellite imagery of their farms to estimate the number of days in living cover that farm has. We're talking big farms out in the Midwest, big row crop farms. And you can actually get a pretty good estimate of the amount of carbon you're sequestering on your farm from your days in living cover, because the carbon that's moving into the soil is mostly moving into the soil through the process of photosynthesis, right, of CO2 being converted into carbohydrates, which are carbon-rich sugar compounds. So what we're seeing here is mostly living cover, which is really good. So we have about 95% living cover. A good trick for estimating percentage of different components in a 30-inch hoop is that a hand fully outspread is about 6% of a 30-inch diameter hoop. My hands are pretty small, so I usually estimate more like 5% for my hand. If you don't spread your fingers, that's closer to 4%. The side of your fist, so like thumb side down, which is a little hard to do, but like that, is supposed to be about 1%, and a thumbprint is 1/3%. So hopefully that's helpful if you need visual estimating tools. The other thing you want to record in your data sheets, which we will provide a link to be able to access the whole manual and data sheets for these tests, is the species richness or species diversity inside the hoop. So looking here, what I'm seeing are chickweed, vetch. This is some kind of an aster. It looks like something in the daisy family. There's also a wild onion. So I would say that this is primarily vetch, probably about 90% vetch if we're thinking about it from a canopy dominance perspective, with maybe 7% chickweed visible, and the remaining 3% is a mix of these asters and wild onion. The importance of the species diversity really has to do with the fact that different soil microbes partner with different plant families. So part of increasing soil life and increasing soil carbon is about increasing the life in the soil, and you can support more life in the soil if you have a more biodiverse soil ecosystem. An ecosystem that is dominated by one plant is only going to support a more limited set of soil microbes. So that's part of why species richness is important, and why cover crop cocktails are a really, really good strategy for increasing soil health. And at this point we can say that the soil temperature is 59 degrees.